Mendel's law of genetics has been a fundamental cornerstone in the study of heredity for over a century. However, recent discoveries in the field of molecular genetics have led to the extension of Mendel's laws to incorporate more complex genetic mechanisms. These new findings highlight the intricate interplay between environmental factors and genetic traits, providing a more nuanced understanding of how traits are inherited. In this report, we will discuss the extension of Mendel's laws and its implications for the future of genetic research. So the reporter are the group 5 and group 6. Unlike the P traits that Mendel examined, most human characteristics do not fall neatly into just two opposing phenotypic categories. So when we say phenotypic, this refers to the individual's observable traits such as hair color, eye, height, and blood type. But it's not always the physical traits of an organism, but behavior is also considered as phenotype. Lentils or lens culinaris provide a graphic illustration of this variation. So lentils are grown in many parts of the world as rich source of both protein and carbohydrates. So the mature plant sets a fruit in the form of diminutive pods that contain two small seeds. Lentils come in an intriguing array of colors and patterns. So the the growers or the producers try to cross between pure breeding lines of lentils, and they come up with a resulting um, surprises. For example, a cross between pure breeding tan and pure breeding gray parents, for example, yields an all brown F1 generation. So when these hybrids self-pollinate, the F2 plants produce not only tan, gray, and brown lentils but also green. Geneticists subjected many kinds of plants and animals to controlled breeding tests using Mendel's 3.1 phenotypic ratio as guidelines. So if the If the traits under analysis behaved as predicted by Mendel's law, then they were assumed to be determined by a single gene with alternative dominant and recessive alleles. Many traits, however, did not behave this way. So for some, no definitive dominance and recessiveness could be observed. So wala siya dominance and recessiveness nga naobserbahan. Next is more than two alleles could be found in a particular cross. So, more than two na ay duha or more than nga alleles nga naa sa particular cross. Others turned out to be determined by two alleles. So, duha ka genes ang makit-an. And other traits were multifactorial. When we say multifactorial, it involves or dependent on a number of factors or causes. So, simple extension Mendel's hypothesis can clarify the relationship between genotype and phenotype. So, when we say genotype, it is an organism's full hereditary information, while phenotype is an organism's actual observed properties such as morphology, development, or behavior and the consequences thereof. Extension to Mendel for single gene inheritance. So, dominance is not always complete. First, dominance is complete when the hybrid resembles one of two pure breeding parents. Second, dominance is incomplete when the hybrid resembles neither parents. Its novel phenotype is usually intermediate. Next is codominance occurs when the hybrid shows traits from both pure breeding parents.
Different dominance relationship. The phenotype of the heterozygate defines the dominance relationship between the two alleles for the same genes, here A1 and A2. Dominance incomplete where the hybrid responsible one of the two pure breeding parents, and the dominance is incomplete when the hybrids responsible neither parent is novel. Phenotypes is usually intermediate, and the other is codominance is occur which the hybrids low trait from the both proved breeding parents. They are the three types of dominance, which is the incomplete, complete, incomplete, and dominant. Ang complete is the naagyo siya makapareha. Kung dili siya makapareha sa A1, makapareha siya sa A2. And sa incomplete, wa yu siya kapareha. Wala yu siya kuan sa A1 and then sa A2. Malahi yang resulta pagka result na. And the sa dominance is sa nag-combine silang duha, ang A1 ug A2. Mo nang pagka-result is the combine the result of each other.
A gene may have more than two alleles. Mendel analyzed other or traits controlled by genes with two alternative alleles. But for many traits, more than two alternatives exist. Here we look at three such traits: human IBO blood types, lentil seed coat patterns, and human histocompatibility anti-cheats. AB blood types. If a person with blood type A meets with a person with blood type B, it is possible in some cases for the couple to have a child that is neither A nor B nor AB, but a fourth blood type called O. ABO blood types are determined by three alleles of one gene. A. 6 genotypes produce the 4 blood group phenotypes. B. Blood serum contains antibodies against foreign red blood cells molecules. C. If a recipient's serum has antibodies against the sugars in a donor's red blood cells, the blood types of recipient and donor are incompatible, and coagulation of red blood cells will occur during transfusion. In this table, a plus indicates compatibility and a minus indicates incompatibility. Antibodies in the donor's blood usually do not cause problems because the amount of transfused antibody is small.
Lentil seed coat patterns. Lentil has another set of examples for multiple alleles. A gene has a seed coat has 5 alleles which are called the spotted, dotted, the clear or the pattern is absent, and the 2 types of marbled. These lentil seed coat patterns are determined by the different alleles of the same gene. Reciprocal process between pairs of pure breeding lines of all patterns classify the dominance relations of all possible pairs of the alleles to reveal a dominant series in which our alleles are listed in order from the most dominant to the most recessive. If the lentil seed coat pattern alleles yielded a 3:1 ratio in the after generation, it indicates that these lentil seed coat patterns are determined by the different alleles of the same gene.
Next is the histocompatibility in humans. So, it refers to the compatibility of cell surface molecules known as histocompatibility antigens. So, these antigens encoded by 3 major histocompatibility genes. This is the HLA-A, HLA-B, and HLA-C in humans. So, it plays a crucial role in the immune response by distinguishing between self and non-self cells. So, each of these genes has a large numbers of alleles resulting in wide range of possible combinations and phenotypic variations in cell surface molecules. So, the extreme variation in histocompatibility antigens has significant medical implications when individuals have different histocompatibility antigens. So, their immune systems can produce antibodies against these non-self antigens. This immune response can lead to the rejection of transplanted organs. This is to minimize the risk of rejection. So, the doctors aim to match the histocompatibility antigen types of organ donors and recipients as closely as possible. So, that is why the family members are often preferred as an organ donors because they are more likely to share HLA alleles due to their closer genetic relationship.
Mutation. Mutation is a change or alteration of the genetic material. This is the changing of the structure of a gene, resulting in a variant form that may be transmitted to subsequent generation caused by the alteration of single base units in DNA, or the deletion, insertion, or arrangement or larger section of genes or chromosomes. So mutation is a changes in a gene sequence, can result in mutant alleles that no longer produce the same level or type of active product as the wild type allele.
Okay, so let's talk about allele frequencies and monomorphic genes. What is allele frequency? So, allele frequency, it refers to the percentage of the total number of gene copies. So, it is a measure of how common or rare particular allele is within a given population. To understand the allele frequency, we have the wild type allele versus mutant allele. So, let's have the wild type allele. So, wild type allele refers to a gene, strain, or a characteristic that prevails among the individuals of the natural population. It contains the most frequently occurring phenotype in the natural population. It is designated as plus sign. So, the example of the wild type allele is the wild type tigers have orange fur with black strips. So the common that is the common population of the wild type tigers. So the difference of the mutant allele is the mutant allele is refers to an individual who is physically different from the others of the same population. So, may kaibahan siya sa iisang pop sa iisang population. So, the phenotype of the mutant allele occurs in lesser frequency in the normal population. So, the mutant allele designated as minus sign. So, the example of the mutant allele is the mutant tigers have white fur with or without black strips. So, that is the wild type allele and the mutant allele. So, yun ang kaibahan nila.
Good morning, everyone. I am Joanna June Ramirez, and my assigned topic is monomorphic and polymorphic. And the monomorphic, a gene with only one common allele frequencies and polymorphic genes. So in monomorphic, the example there is male and female penguins are very similar in appearance and can be impossible to distinguish. So in penguins, the male and the female is very similar. So, maglisod ta kung asa ang laki o bahi kay very similar, and that is the example of monomorphic. The polymorphic is genes have more than one common allele. So, the example there is the blood groups. The blood groups, a gene have more than one common. So the blood groups. All the types of blood groups are example of genetic polymorphism.
So why sickle cell disease is a result of faulty hemoglobin results? So, sickle cell disease is a result of faulty hemoglobin due to the production of abnormal hemoglobin S protein, which leads to the characteristic thickening of red blood cells and the associated symptoms of the disease. So which is alpha and beta? So alpha globin is a protein that consists of two identical alpha chains, and it is a component of the hemoglobin molecule in red blood cells. So ang alpha globin mao responsible nga mo-carry sa oxygen from the lungs to the tissue in the body. So the most common mutant allele of beta globin is the HBS allele, which result from the single nucleotide substitution in the HBB gene. This mutant causes a change in the amino acid sequence of the beta globin protein.
Hey everyone, I am Santiago Milas Jr. and this is my report 3.1, extension for Mendel single gene inheritance. Recessive lethal alleles. Significant variation of phylotropy occurs in alleles that not only products a visible phenotype but also affect viability. Delayed lethality. Homozygotes may survive beyond birth and die after the reduced consequences of the genetic defect. Example: humans infant with Tay Sachs disease. The seemingly normal newborn remain healthy for 5 to 6 months but then develops blindness, paralysis, mental impairment, and other symptoms of a deterioration nervous system. So, ana day manala siya kuan normal the newborn is remain healthy but 5 to 6 months is maka-develop siya mga blindness, paralysis, and mental impairment and other symptoms due to deteriorating in their nervous system. That's all my report, sir. Thank you.
Right, so here now, I will give you an overview of what sickle cell disease. Okay, so sickle cell disease is the name of four group of inherited health condition that affect the red blood cells. Just like for example, most common serious type of disease is called anemia. So sickle cell disease is serious and lifelong health condition, particularly with Caribbean or Africa family background. So
Sir, good day, sir. So I am Danny Daguya Ibib and my partner Jandris Ubag. So our assigned topic is all about one gene may contribute to several characteristics. So extension to Mendel laws. So Mendel derived his laws from studies in which one gene determined one trait, but also the careful observer he himself noted possible departures. In listening the traits selected for his PIA experiment, Mendel remarked the specific seed coat colors are also associated with specific flowers colors. The phenomenon of a single gene determining a number of distinct and seemingly unrelated characteristics is known as pleurotropy. So, example. So, many men develop respiratory issues and are also sterile. These men are said to exhibit a syndrome. So, syndrome is a group of problems that are usually seen together. So, researchers have found that the fault lies with the recessive allele of a single gene. So, and then the gene's normal dominant allele specifies a protein necessary for the actions of cilia and flagella. So both of which are hair-like structures extending from the surface of some cells. So and then we have delayed lethality. So in the preceding discussions, we have a describes recessive alleles. So that result in the death of homozygotes prenatally, that is in utero. So with some mutations, however, homozygotes may survive beyond the birth and die later from the deleterious consequences of the genetic defect. So, for example, human infants with Tay Sachs disease. So, the seemingly normal newborn remain healthy for 5 to 6 months. And then, but the develops blindness, paralysis, mental impairment, and other symptoms at the deteriorating nervous system. So, then, the approximate incidence of such size Tay Sachs among liverts is 1/35.00 worldwide. Millions worldwide. But it's 1/3,000 among Jewish pupil of Eastern European descent. So, that's all, sir.
So, good day, sir. I'm John Reyes Ubag, together with Danny Ibib. So, in continuation for our topic, one gene may contribute to several characteristics. So, recessive lethal alleles. Significant variation of pleiotrophy occurs in alleles that not only produce a visible phenotype but also affect viability. So, Mendel assumed that all genotypes are equally viable. That is, they have the same likelihood of survival. If all genotypes were not equally viable and a large percentage of homozygotes for a particular allele died before germination or birth, you would not be able to count them. So, figure 3.8, a to the power of y is a pleiotrophic and recessive lethal allele. So, A, all yellow mice are heterozygotes. So, a cross between inbred agouti mice and yellow mice yields a 1:1 ratio of yellow to agouti progeny. The yellow mice are therefore a to the power of y, a heterozygotes, and for the trait of coat color, a to the power of y for yellow is dominant to a for agouti. So, figure B. 2 copies of A to the power of Y cause lethality. So, yellow mice do not breed through. In a yellow by yellow cross, the 2:1 ratio of yellow to agouti progeny indicates that the A to the power of Y allele is a recessive lethal. So, from this result, we can draw 3 conclusions. 1. All yellow mice must carry the wild type A allele, even though they do not express the agouti phenotype. 2. Yellow is therefore dominant to agouti. Lastly, 3. All yellow mice are A to the power of Y A heterozygotes. So that's all. Thank you.
Good afternoon, everyone. I am Emmanuel. I am report on one in my own duty to survive is so I am looking benefit by me working on in a number I is someone many men ignore men a no a fan is your eye also is our table as a only of them I think of it for it is not of I also affect player